Hi, everyone. Thank you for joining the third and last webinar in our three-part series on using in silico and in vitro approaches for next generation risk assessment of potential respiratory toxicants. This series is being co-organized by the U.S. Environmental Protection Agency, PETA Science Consortium International, Syngenta, and Unilever. My name is Marie Hargrove. I'm currently a technical expert at Syngenta Crop Protection, and I'll be co-moderating today's webinar with Gino Scarano, who is a toxicologist at the US EPA, and Andrea Stuckey, Science Advisor at PETA Science Consortium International. We have three presentations today showcasing how in silico and in vitro methods can be used to help risk assessment. First, we'll hear from myself, followed by Maria Baltazar and Iris Muller from Unilever. The last presentation will be given by Sandra Verstralen and Evelyn Friends from the Flemish Institute for Technological Research, Vitro. I'd like to point out that the slides and a recording from today's webinar will be posted at the link on the screen shortly after the webinar. You will also find the recordings from the previous webinars on that webpage. There will be time for questions for all three speakers at the end of the last presentation. Everyone is on mute, but you can type any questions you may have in the question section within your GoToWebinar toolbar, which should be on the right-hand side of your screen. If you send in a question or a comment, please indicate if it is for a specific speaker or for everyone. I now turn over to Gino Scarano for his introductory comment.
Thank you, Marie. My name is Gino Scarano, and I'm a senior advisor in the New Chemicals Division in the Office of Pollution Prevention and Toxics here at the US EPA. EPA is proud to be a co-organizer of this three-part webinar series, along with the PETA International Science Consortium, Syngenta, and Unilever. I want to thank the organizing committee for pulling all this together, especially Andy and Amy from the PETA International Science Consortium. So welcome all, and I look forward to the presentations and invigorating discussions. So now I'd like to introduce our first speaker, Marie Hargrove, who's also a co-moderator. Dr. Marie Hargrove is a technical expert within the Human Safety Group at Syngenta Crop Protection. A graduate of the Pharmacology and Toxicology program at East Carolina University, Dr. Hargrove completed a postdoctoral fellowship at the EPA National Health and Environmental Effects Research Laboratory within the Environmental Public Health Division prior to her current role at Syngenta. She has directed and managed numerous in vivo toxicological studies which characterize respiratory and systemic effects of known criteria air pollutants in models representative of susceptible populations. She currently focuses on managing studies in systemic toxicology and endocrine disruption, which ultimately supports registration of plant protection products via human health risk assessment. Marie?
Thank you, Gino, for the lovely introduction. I'd like to thank the organizing committee. First of all, let me fix this screen here. Let's see. Sorry for the technical difficulty this morning.
Marie, would you like that I share my screen?
Marie, if you go what you were doing, putting in presenting mode, and then if you go to the display and change it to swap presenting, that usually works with me.
Let me see.
And,
Thanks, Marie.
oh, no, it seems.
There you go. Oh, yeah.
Yeah,
now
it's not giving me the option. So since
okay.
I've wasted a few minutes, Andy, do you mind sharing the slides?
Yes, I can do that.
Thank you.
Um, do you see my screen now?
Yes, thanks so much. Thank you for your patience, everyone. My apologies. Okay, so I'd like to thank the organizing committee for the opportunity to give this talk this morning, and to the audience. I thank all of you for attending, whether it's early morning or late afternoon. We greatly appreciate it. So the title of my talk today is Refining Inhalation Risk Assessment of a Contact Irritant Using Computational Fluid Dynamics and the Mucilair Airway Assay. So I will admit, when creating this title, I neglected a couple aspects of this approach. So I hope you will appreciate that I have included some other facets of the NAM approach that we used in this talk today. So Andy, next slide, please. Okay, so an overview of the talk. So I like to start with a brief introduction or overview here. And so the intro will be followed by just briefly explain the evaluation framework for NAMs for human health safety assessment. And the majority of the talk will focus on the chlorothalonil case study and these different portions of that, the pesticide exposure data, the human CFD simulation, the use of the Mucilair assay, as well as bench dose modeling methods, followed by the conclusion and the acknowledgments. Next slide, please. So numerous agrochemicals need to be assessed in the context of regulatory authorities for pulmonary toxicity. So on the right-hand side of the slide, there's a risk characterization Venn diagram with risk assessment on the left and risk management on the right-hand side. So today we're going to just focus in on that red circle containing risk assessment. So the toxicology of air pollutants and chemicals has relied on in vivo and in vitro testing for a number of decades. And so there's a high demand to find and implement new approaches that encompass the 3R principles, which are replacement, reduction, and refinement. And so in inhalation toxicology, the need for non-animal alternatives has given rise to several different types of assays that are commercially available. And more importantly, health effects due to inhalation of substances are therefore of considerable interest to many and form the basis for the use of development of these new approach methodologies. Next slide. So many international chemical management policies and laws have escalated the demand for data on the safety of chemicals. And so to meet this demand, a variety of new data streams in hazard, exposure, and dose evaluation are being considered to augment traditional toxicology data, which has mostly relied on animal models. So this slide contains a couple snapshots of recent papers looking at human health risk assessment, as well as snapshots from ECHA as well as the US EPA. So in the United States, the recent Frank Lautenberg Chemical Safety for the 21st Century Act amended TSCA by including, among other things, the promotion of the use of alternatives to vertebrate animals under the Chemical Testing Authority. So at this point, this is the third webinar. So if you are familiar with NAMs, this is repetitive to you, but if not, NAMs, the broad statement includes in silico and chemical and in vitro assays, as well as the inclusion of information from the exposure of chemicals in the context of hazard. So NAMs for toxicity testing, including these alternatives to animal testing approaches, we hope may be able to provide a large amount of data and fill in information gaps for both hazard and exposure. Next slide. So a fitness for purpose evaluation process must be able to match the nature and the speed of scientific advancement while building confidence in the acceptability of these NAMs. And so how do you do that? So on the left-hand side, it's just kind of a step-by-step view of how to establish a fitness for purpose or establishing fitness for purpose of a NAM, starting with the explanation of mechanistic basis to regulatory acceptance. So there's a recent publication, the reference is at the bottom of the slide, explaining this framework. So these new approaches to establish confidence in NAMs are being developed based on human biology and mechanistic relevance, rather than an empirical approach based on apical effects in animal studies for evaluating performance. So there are 3 steps. So step 1 is determining the context of use. So you have to decide whether or not this assay will be used for prioritization, hazard screening, or risk assessment. The second step is addressing the core principles, accuracy, transparency, understanding limitations, as well as domain of applicability. So the combination of steps 1 and 2 form your problem formulation foundation. And then the last step is the fitness for purpose criteria. So in step 1, you decided where this assay will fit or what purpose it will serve, and that determines the steps of the criteria that you must follow. And so that table illustrates, the table on the right hand side illustrates the steps for that. And this can be viewed as a means of validation, but not in the traditional sense. Importantly, this criteria is dependent upon how you use the assay. As you go from left to right, you'll notice there's a sliding scale there in the table. More steps are required to meet the criteria. Next slide, please. So I mentioned the evaluation framework, the problem formulation foundation. So we at Syngenta created this problem statement. So it's as follows. A new approach method can be developed and will be suitable to inform inhalation toxicity in lieu of a subchronic whole animal inhalation study. Next slide, please. So I've mentioned the problem statement. I've mentioned the problem foundation, problem formulation foundation. And so what exactly was the problem? And in reference, and just to provide a little bit of background on what chlorothalonil is. So during pesticide re-registration, the agency examines the completeness of the tox database. So to determine if it can still support risk assessments for safe use. And so a repeat dose 90-day inhalation study is considered a regulatory requirement. It was requested by the US EPA. However, no new or additional data will be forthcoming from such a study that will improve safety assessment, and furthermore, no additional systemic risk from inhalation exposure because chlorothalonil does produce contact irritation in the respiratory tract. So what is chlorothalonil? It's a broad spectrum non-systemic pesticide mainly used as a fungicide to control foliar diseases on a wide variety of vegetables, field and ornamental crops. It also has non-ag uses. It has a long history of safe use since 1966. It is known as a contact irritant inducing respiratory tract effects in acute studies. And so we began to think about, well, how will we address this regulatory requirement? So it's been quite the journey. So the re-registration process began in 2010, and then I'm just going to skip ahead there. The NAM development for this started in 2014. So today I have the honor of presenting to you a combination of alternative approaches conducted to define chlorothalonil's inhalation toxicity, which include the CFD modeling, the use of the Mucilair, looking at pesticide exposure data, as well as bench dose modeling. Next slide. So at the top of the slide, you'll notice the traditional inhalation model, which involves inhalation exposure via aerosols in rodents or some other animal model that provides information to calculate the human equivalent concentration, otherwise known as the HEC. And the HEC allows for risk characterization. So at the bottom half of the slide, the model that we chose to use is a human relevant exposure-based inhalation model. And so this NAM involves looking at particle size distribution, or PSD. And for in vitro testing, we use the Mucilair three-dimensional assay and a dosimetry model, which is the CFD modeling to mimic the human upper airway tract, along with calculations for BMD levels and looking at pesticide exposure data to identify safe operator use to the pesticide chlorothalonil following field application. So this NAM replaces the traditional approach of using animals to calculate HEC, which is used in risk assessment for those using chlorothalonil containing formulations as well as sprays. Next slide. So one of the first steps was to look at pesticide exposure data. So potential inhalation exposure of agricultural workers and bystanders to aerosolized particles emitted by typical agricultural spray nozzles is influenced by particle size distribution of the spray. However, inhalation risk assessments do not currently factor in the human relevant PSD that may be inhaled during these types of pesticide handling activities. So this study was conducted to characterize the PSD of the aerosols, and these aerosols were collected in these OSHA versatile sampler tubes that are pictured on the right-hand side of the slide on this person that's working. The slide was, the side-by-side air sampling with the OVS and a Respircon TM particle sampler was conducted to categorize the size distribution of the aerosols collected on these tubes during the spraying of a diluted formulation. And so this information on the PSD of pesticide applications is very important because it's used to refine inhalation risk assessments for agricultural workers as well as bystanders. Next slide. So a little bit more detail about exposure. So the human respiratory tract, all of you, I suspect, on this webinar are quite familiar with this and how it works. So this is just a general idea of how the human exposure piece. So you have aerosols that deposit in various portions of the respiratory tract. And so in the upper respiratory airways, you have a higher deposition of coarse particles that range from 2.5 microns to 10 microns, which includes dust, mold, pollen, and crustal material. The bronchiolar airways has a higher deposition of fine particles, and those fine particles consist of combustion particles, organic compounds, as well as metals. And then last but not least, you have the deposition of nanoparticles or ultrafine particles. So from a general sense, the fine particles, if you have a single human hair, which is about 70 microns, is 30 times larger than the largest fine particle. Next slide. So in order to categorize or in order to utilize the in vitro data for the human health risk assessment, we had to use a dosimetry model to calculate external concentrations that would produce the surface concentrations of deposited chlorothalonil in each region of the upper respiratory tract. So these models are used to determine internal doses of a chemical and provide information that aids in the understanding between the external exposure and a biological response. So each simulation of the CFD model was based on a 68 kilogram male subject, assuming 1 mg per liter aerosol concentration, a minute volume of 7.4 liters per minute, and a breathing frequency of 20 breaths a minute. So since the CFD since the CFD model is essentially generating results for a generic water droplet, the nasal and oral CFD deposition results are considered chemical agnostic. So for the NAM approach, Syngenta predicted deposition of chlorothalonil in site-specific regions of the respiratory tract, which are featured here. And so what we found is that the percentage is laid out. So 99.7% is deposited in the nose, the pharynx, and the larynx, and a very small portion, 0.007%, is deposited in the remaining airways, and 0 doesn't escape the 3D lung, and 0.3 remains suspended in the airways at the end of inhalation. So the resulting computational model provides the necessary common denominator of localized tissue doses critical to integrating data and defining appropriate comparative dose metrics within this source to outcome risk assessment approach. Next slide. So I mentioned the pesticide exposure data looking at the characterization of particle size distribution. We also did the CFD modeling, but you also have to keep in mind of the data that you currently have in your database, right? So this is a table illustrating the acute effects in the male and female rats. Furthermore, the chlorothalonil-induced pathology in the rat after inhalation exposure. So as you can see in the table, the acute exposure of chlorothalonil at 2, 4, and 6 hours is similar when you look at both inflammation and epithelial necrosis and ulceration. So this was the justification we used with the US EPA for the use of the Mucilair assay, indicating that it is representative in vivo, and the agency agreed. So chlorothalonil, as classified by the US EPA, is in toxicity category 2 following acute 4-hour exposure, being an inhalation route. So an adequate in vivo subchronic inhalation study was not available for chlorothalonil. And so the alternative in vitro approach may be taken when you don't have this in vivo data, when it's not adequate or it's just not available to establish a toxicity endpoint for inhalation risk assessment. Next slide. So what chlorothalonil concentration in the aqueous layer covering the airway epithelium is necessary to damage the epithelium underneath? So as we began to explore which method and got the okay to use the Mucilair, you start to think about, well, what endpoints do I want to look at in the assay? So at the top, as you look at this, the top image with the arrow pointing down from chlorothalonil, it's a normal pseudostratified epithelium, and then beneath that, where it says hydrolysis and metabolism ROS, it's been exposed to chlorothalonil, producing this squamous metaplasia, which is a normal response to irritation. And on the bottom right-hand side of the slide, it indicates the endpoints that we wanted to think about evaluating with the Mucilair model. Next slide. So if you are familiar, this is repetitive, but if not, hopefully I can provide you a brief background on the Mucilair assay. It's a 3D model of human airway epithelium formed from differentiated human cells. And so an in vivo study would only show the tox effects at the target tissues in the respiratory tract, but not the target tissue exposure concentration that caused the effect. So the Mucilair system was identified as the optimal in vitro model to assess damage to respiratory epithelial cells caused by exposure to chlorothalonil. So the Mucilair, these cells can be collected from healthy donors, and these cells are cultured on CoStar transworld polyester membrane units using an optimally defined media. And so you can choose from a single donor or a pool of donors in the nasal healthy version. It has different anatomical sites. You can choose patients with several pathologies, from COPD to those that are identified as smokers. It's quite easy to use, and it also allows for performing acute and long-term as well as repeat dose studies or efficacy testing of solid chemicals and mixtures. So on the right-hand slide is just a comparison of Mucilair against the other commercially available options. Next slide. So the Mucilair endpoint parameters that we chose to look at are TEER, transepithelial electrical resistance, LDH, lactate dehydrogenase, and rizazolam metabolism. And so at the bottom of the slide, on the left-hand side, it shows the information that you can obtain from using the Mucilair. In the middle, it's just a caption of the 24-well plate, and on the right-hand side is the electron microscopy showing the configuration at 10 and 50 microns. And if you're interested in seeing the other capabilities of this assay, you can go to the epithelium's website. So these parameters, a decrease in tear, if you observe a decrease in tear, it's indicative of a decrease in integrity of tight junctions between cells in the membrane. LDH is indicative of cellular cytotoxic membrane damage. And then rizazolam metabolism or rizazolam reduction is indicative of metabolic competence. So if you observe a decreased fluorescence, it's an indicator for a decrease in metabolic competence. Next slide. So I shared with you the endpoints that we looked at. We did not look at histology in this particular experiment, but in subsequent experiments, we did add this on, in case you were wondering. And so the tissues were taken from 5 healthy donors. We chose a 24-hour exposure. Chlorothalonil was applied as in formulation form. So we used 10 concentrations per donor and 6 replicates per concentration per donor. And so the concentrations that we use are reflected in the table on the right-hand side. So we chose the 24-hour exposure as a conservative approach. We also used 5 separate donors to evaluate variance. So in subsequent studies, we used the pooled donor to have a representative approach. But in this case, just to start, we chose to go with the 5 healthy donors just to be sure there was little variance between them. Next slide. So benchmark dose modeling. So these benchmark dose methods are used by the US EPA and throughout the world for dose response analyses to support chemical risk assessments and actions. So a benchmark dose is a dose or concentration that produces a predetermined change in the response rate of an adverse effect. And so it's less dependent on dose selection and spacing and takes into account the shape of the dose response curve as reflected in the image on the right-hand side. And so this tool was developed by the EPA, and the software is located on the EPA website. Next slide. Okay, so what we did here is with the chlorothalonil Mucilair study results, what we did find is that the individual donor responses are quite similar across the 5 donors, which is good, and suggests that inter-donor variability and sensitivity is low, right? And so all of the endpoints responded similar to chlorothalonil in vivo, which is what we anticipated. And so so what we have here in the tables, on the left-hand side is the BMDL measured in mg per liter, and then on the right-hand side is BMDL in mg per centimeter squared. So for Syngenta's BMD analysis, the doses were log transformed and fit with a modified Hill model. The agency performed its own BMD on the untransformed data for comparison and found the Hill model to be the best fit for the data. So for both models, they were fit the data well visually, and AIC values indicate the relative fit for a model for this particular data set. So overall, the untransformed data provided similar or lower AIC values than the transformed, and however, the BMD and the BMDL values obtained were lower and considered protective. And so what are those values? So on the left-hand side, you'll notice that the range for this was in 40 to 125 mg per liter, which was then translated using the equation at the bottom of the slide to mass per unit surface area. And so an overall BMDL value of 0.00730 mg per centimeter squared was established as the inhalation toxicity endpoint for chlorothalonil. Next slide. So I presented a lot of information today. I hope this does not overwhelm you. So one of the first pieces was characterizing the particle size distribution combined with the Mucilair assay, as well as the dosimetry model, the CFD modeling, and all of this is used to calculate the human equivalent concentration, which can be used to inform or categorize the risk. Next slide. Okay, so what does this all mean, or collectively, what does it mean? So on the left-hand side, the residential bystander risk assessment was conducted based on the traditional methodologies. And on the right-hand side, the risk assessment was conducted based on the combination of the NAMs that I just presented. And as you can see, we're still within the green portion of the graph, meaning this is acceptable. And we're quite excited about it. And so the manuscript is referenced at the bottom of the slide and currently under review. Next slide. Okay, so the outcome of all of this work, it addresses the inhalation study requirement, the point of departure, the uncertainty factors, and this has been a long journey for Syngenta, and I'm very fortunate to be able to present this data to you today. As this proposal began in 2014, when it was presented to the US EPA, who recognized the value in the proposal for chlorothalonil as well as other contact irritants, and supported the movement of a NAM in lieu of an in vivo laboratory in the lieu of animal testing. So the EPA encouraged further development and determined that external peer review and public dialogue will be needed to before applying this approach to human risk assessment. And so a preliminary risk assessment and issue paper was then published by the EPA in 2018. And as featured on the right-hand side, there was a FIFRA scientific advisory panel that used chlorothalonil as a proof of concept, and they also provided their support as well as advice on how to move this approach forward. And so the final report came in 2019, and I'm very happy to share that all of this data was generated and used for a formal risk assessment, and we've now received that draft risk assessment about 2 weeks ago. So it's been quite an exciting journey and in learning more about NAMs and how they can be used in risk assessment. Next slide. All right, lots of conclusions and key learnings here. So what do we know? So chlorothalonil was under reg review by the EPA, and a 90-day inhalation study was required for registration. The goal was to develop and demonstrate that an in vitro assay combined with an in silico model was fit for purpose as a new approach method. For respiratory irritants such as chlorothalonil, we now know an alternative in vitro approach, such as the Mucilair, may be taken when in vivo data are not adequate or not available. So the characterization of the PSD of aerosols was provided useful information to help refine inhalation risk assessments for ag workers and bystanders. And using the 3 endpoints, we were able to calculate a BMDL level of 0.00730. And this information, combined with the information obtained for the CFD modeling, which is not reflected on the slide, was useful in calculating the human equivalent concentration to inform inhalation risk assessment. And so with the application of this NAM to refine the inhalation risk assessment for chlorothalonil, the recommendation has been to waive the 90-day inhalation toxicity study for chlorothalonil, which saves time, resources, and several animals. So we're very excited about that. And overall, I did notice there's one mistake in the very last line. The US EPA is using the Mucilair assay to inform intermediate inhalation risk. And with that, I thank you so much for listening to this talk. Very last slide, Andy, next slide, please. I'd like to acknowledge all of these wonderful scientists and team for their hardworking efforts, and I'm very thankful to be a part of this talk today and present their data. Thank you.
Thank you, Marie. As a reminder, we will hold all questions to the end, but please send them in as you have them using the GoToWebinar questions panel, and we will ask them after our last presentation.
Speaking next will be Drs. Maria Baltazar and Iris Muller. Maria is a science leader at the Safety and Environment Assurance Center at Unilever. She has a background in pharmaceutical sciences and a PhD in toxicology from the University of Porto in Portugal and has been a toxicologist in industry for 7 years. Maria leads the inhalation capability platform and projects leading to advanced next generation risk assessment tools for systemic toxicity at Unilever. Dr. Iris Muller is a science leader at the Safety and Environment Assurance Center at Unilever as well. She has a background in molecular cell biology and is leading the in vitro capability area of the inhalation platform, building an inhalation toolbox which can be used for next generation risk assessment. In addition, she is also co-leading the DART project in Unilever. Before working for Unilever, she worked at the German Federal Institute for Risk Assessment.
Thank you very much for that kind of introduction, Marie. So Iris and I will be talking about our ongoing development of a next generation risk assessment framework for inhalation safety of consumer products. First, I would like to just give a quick introduction to our industry. Unilever is a global fast consumer goods company that makes multiple brands across 3 different spaces, beauty and personal care, home care, and foods and refreshments. Our department, the Safety and Environmental Assurance Center, is a team of safety and environmental sustainability scientists. We use the latest techniques, deep science, scientific expertise, and an evidence-based approach to ensure that our products are safe for consumers, but also for workers and for the environment. Safety assessments of ingredients in products have relied on apical endpoints derived from animal testing. However, ethical and regulatory considerations on animal use, for example, in Europe, we have an animal testing ban for cosmetics that has been extended also for other countries. In addition to the scientific need to use more human relevant data, have led to the emergence of this next generation risk assessment, so NGRA. And NGRA is defined as an exposure-led, hypothesis-driven risk assessment approach that integrates new approach methodologies and NAMs to ensure safety without the use of animal testing. And there's been a lot of progress, and I just put here some examples of the different reports that have been issued over the years, and showcasing how we can now use the 21st century science to really improve our risk assessments. So how does NGRA look like in practice? So we try to demonstrate the principles of NGRA outlined, for example, by other external bodies like the ICCRs, the International Corporation on Cosmetics Regulation, and also by the US EPA, where they outlined in their blueprint the strategy. So to this end, to demonstrate kind of several principles have been outlined by these bodies, external bodies, we published an hypothetical safety assessment of human cosmetics products. For this, we created this framework that we show here that guided us in the risk assessment process from the initial problem formulation to data generation and finally to a safety decision. And we have published that recently. Briefly, just to give an example of kind of our approach, for this systemic toxicity case study, internal concentrations like as plasma Cmax concentrations were estimated using a physiological based kinetic model for dermally applied coumarin, and systemic toxicity was assessed using a battery of in vitro NAMs to identify points of departures for a variety of biological effects, such as receptor-mediated and immunomodulatory effects, but also for less specific effects using tools like cell stress panel and transcriptomics. So these PODs in the end were then compared to the calculated in vivo exposure, and to calculate then this margin of exposure associated with uncertainty. So you can see on the graph on the right, that comparison of the point of departures with the estimated plasma Cmax in yellow. And this approach really allowed us to make the safety decision and demonstrates those principles also outlined by the EPA of protection and not prediction. Whereas if we don't observe any biological activity at the predicted exposure concentration, then there cannot be an adverse effect. And we're now exploring this approach and evaluating this approach with a large number of chemicals with a wide range of modes of action to test the hypothesis that these tools are protective of human health. And here on the slide, I also have some of the key references that also have been actually included recently in the new notes of guidance from the SEAC, which is the cosmetic regulator in Europe, and it has included some of our work. And I just wanted to give this introduction about this general approach to bear in mind, because this is the same mindset that we'll try to apply for inhalation toxicity. So our risk assessments are always exposure driven, and therefore an inhalation risk will be triggered depending on the product type that we are risk assessing. And here on the slide are some examples of the type of products, the range of type of products we tend to risk assess, and they are used quite differently, from each other. Some are applied to the hair, some are applied to the axilla, underarm, and some are applied to the kitchen or to bathrooms as cleaning products. So we use robust data on how our consumers use our products. We tend to know how they're going to and how frequently they're going to use our products. And we also have information on formulation and hardware, which also has a big impact on the exposure. And after we'll conduct an appropriate appraisal of the potential hazards of ingredients, which kind of leads to my next slide, where we've been trying to put together also an inhalation framework, which we have adapted from our systemic framework that we published using the Cumulin case study. And we've mapped the existing and novel tools that can be used in inhalation risk assessment. So I won't go into a lot of detail, but just kind of giving a general overview. For exposure, we start by identifying, like I was saying, the product type, the use scenario, consumer practices, followed by formulation and particle size and distribution, which kindly Marie already outlined the influence that the particle size distribution has on the exposure. With this information, we can follow a series of tiered approaches to estimate exposure, starting with simple and conservative tools, like the Tier 1 screen assessment outlined in the ECHA guidance, for example, and if necessary, refining with more complex tools like the Consexpo, the RIFM two-box model, and also go into experimental studies as well to refine our exposure. From the hazard side, as any risk assessment, we can start by using Silico tools, performing literature reviews or read across. If there is no existing information and you cannot do read across, then you can use TTC, so the exposure-based weighting approaches. And if none of these cases you cannot use, then you are in that data generation box, that purple box, and in the NGRA space. In the purple, lighter purple boxes, we have the endpoints and systems we've been trying to evaluate in the last few years, and then we'll be focused on the presentation today. And in the grey boxes are the areas for future work where we have just started on to do some exploration and some partners. These are the areas very targeted specific to lungs effects, but any of the tools described in the Cumulin case study could be used to complement the risk assessment depending on the type of chemical we are risk assessing. For example, some of the molecular initiating events present in our receptor panels are very relevant for inhalation toxicity as well. From the in vitro data, then on the next box in green, we would then perform dose response modeling and determine a point of departure and compare to the predicted exposure, just like in the Cumulin case study. But in this case, we have different dose metrics, as we're going to talk about, and the assays are also slightly different. To give you an overview how we've designed our strategy, it was very much in the context of some of the business needs that we face. So we have requests for having new polymers for antiperspirants and some silanes for use in the purpose cleaners. So we did a brainstorm, look at some of the chemicals, what are the chemistry of these chemicals, the potential hazards associated with them, and do we have any existing information. Afterwards, Also looking, so in which formulations and which product types are these ingredients going to be included in. So again, information on particle size distribution, how the consumers are going to use. Then we would, for all these chemicals, will perform this tier modeling approach, and we will use MPBD and the combination with the particle size distribution to guide us where those particles are going to deposit in the respiratory tract, and then select some relevant in vitro exposure doses, but also the systems. So taking all together this information, so the compounds and the exposure, we then brainstorm about what are those key hazards of concern, and then we identify these 4 concerns, so lung fibrosis, impairment of mucociliary clearance, lung surfactant inhibition, and persistency. And in blue are the areas that we have prioritized and will be presenting today. And we've used adapting these endpoints. We selected the cell models that we wanted to study, and that's what Iris is going to focus on in her presentation. And the biomarkers are very much aligned to MIEs and key events of critical AOPs that have been reviewed by others. So some of the publications outlined here. The next, we wanted to evaluate our approach. So our general strategy to NGRA has been using this benchmarking approach. We're doing this in the systemic space and also in our skin allergy strategies. In a nutshell, or what we've been trying to do is for each chemical, we identified exposures which have been associated with either a high or low risk scenario, with a few exceptions. Some of the chemicals here on the screen were used to target a specific effect, like, for example, cilia beating frequency. But from the majority of the chemicals, we try to find in which classification there will be a high risk or low risk and their exposure. And for these chemicals, we then predicted that exposure using the models I described above, and collated evidence from human data primarily, and if that didn't exist, some animal data in combination with a history of safe use to support the classification. So some of those compounds exist in products in the market and without adverse effects. We also binned the chemicals according to the endpoint we wanted to evaluate our systems for, so modulators of cilia beating and mucus production, so the clearance part, inflammation, fibrosis, cytotoxicity, and then some negative controls where we had other data and we had also the history safe use data. So this is quite a lot of very large data set, and we're still processing some of this data. And for this reason, we decided for this webinar to focus on in one of the benchmark chemicals to demonstrate the approach that will be then hopefully applied to the majority of chemicals. So at least we could demonstrate the framework with one chemical. So I'll be talking about polyhexamethylene guanidine phosphate, PHMG, as a benchmark chemical. This is a chemical that we found there was quite a lot of data that associated PHMG use during normal use of household humidifiers was linked to this acute interstitial pneumonia and pulmonary fibrosis in humans. And there was also some data suggesting that PHMG-induced ROS generation increased the production of inflammatory fibrotic cytokines and profibrotic genes in several cell models. So this was used, therefore, that was the reason why we decided to use this as a benchmark chemical. So our first part in our framework is the exposure assessment, and we used the tier 1 assessment because this has been actually described quite extensively also in the literature. So we looked for an exposure scenario that had that link between exposure and adverse effects in humans. We selected the worst scenario describing those in publications. So based on the concentration of PHMG in the disinfectant, then is used in that humidifier to prevent microbial growth. So the concentration, the volume that is added, how frequently people are going to use, and usually the volume of the room where the humidifier is going to be used. And that leads us to the airborne PHMG level level, which is 0.95 milligrams per meter cube. And that is kind of that worst case figure. So now we have the airborne PHMG level, but now we're interested actually in that lung deposition and what is going to be the concentration at the site. So we're not only interested in that inhale amount, so taking into account the breathing rate and the duration of exposure, but we're also interested in the total and regional deposition of the particles. So we can then design the in vitro testing aiming at predicting effects in the likely target tissues of humans. So to this end, we use the MPPD, which has already been talked about in the other series of webinar, which is a mechanistic dosimetry model that facilitates this calculation of regional and site-specific deposition and clearance of particles. For describing the human lung geometry, there are available a few options in the model. You have the 5-lobe PU shum, you have the stochastic, the age-specific symmetric model. But for this case study, we selected the stochastic lung model, and we also calculated the exposure for different percentiles, used the particle MMAD, described also in the literature, so 80 nanometers, and we did the simulations with and without clearance. And then we calculated the dose in micrograms per square centimeter by adjusting the exposure duration, which was 11 hours per day. So that was also something that was described in the literature as that the maximum exposure time. On the next slide, we have then the results for the total and regional deposition based on the consumer exposure scenario and based on the 40th percentile for the various time points, assuming accumulation over time in between exposures. And these time points match the exposure that we'll be showing in the next slides. So now I'll pass over to Iris, who will talk about the rest of the framework for the case study and how we'll integrate exposure with the hazard assessment.
Thank you, Maria. So the lung deposition calculation as just described by Maria, I think I don't have control at the moment.
It said you had control, but I'll give you again. Actually, no, you do. Can you move it now?
No. Can't.
Yeah.
Is it moving? It's not moving for me.
The thing is, it has a tick for you. Maybe I can just move the slide. It has a tick near you. So
Yeah, if you just move it for me, it's also fine.
Can you try again? Sorry. Can you try now? It works in the practice. Okay, maybe I'll move it for you.
Okay, cool. Thank you. So can you move it to the next slide, please? Okay. So the lung deposition calculation, as just described by Maria, is used in two ways in our framework. So first, it is used in the data generation to provide the dose range where we performed our in vitro experiments at. And second, it is used for comparison with the in vitro derived points of departure to define margin of exposure in the context of decision-making for inhalation risk assessment. Therefore, the in vitro concentration response modeling, as here highlighted in red, is an important part of our NGRA framework, and the mathematical model used needs to be fit for purpose to align with the experimental setting, but also to address experimental and biological uncertainties. As mentioned by Maria before, data generation will be driven by identification of key hazard concerns, and therefore we'll try to establish an inhalation toolbox which covers all the endpoints of concerns. The two other boxes here highlighted red in the data generation part of the framework represents identify hazard. We are investigating in a proof of concept study at the moment, and as Maria mentioned, this is the part we want to describe further in the talk. The third box shows some additional endpoints we have already identified and where we have started some collaboration with different partners. We also started to investigate in some more advanced cell systems like organ on the chip or MPS systems, which we will run in a higher tier for our NGRA framework. Both boxes are grayed out because I will not go into detail for that in the talk. For the proof of concept study, we separated out the data generation part into upper airways and lower airways. And this is done to correspond to the cells in vivo and their function, and also to the expected toxic endpoints. Also remembering, we produce this for consumer risk assessment. So we will have like normally quite low exposure, but therefore over a long period of time. So probably some of the products will be used for years. So there we're really interested in acute, but also in chronic responses. Can you move to the next slide? For the hazard detection in the upper airways, we choose the well-studied Mucilair cell system. And there's a lot of literature out there, and also like Marie did a really good job to introduce the cell system. So I will not go into further details. But just as a quick reminder, the cells can be grown at the air-liquid interface, which allows nebulized treatment of the substances, which mimics quite well our in vivo exposure. And also the cells are quite stable for a long time, for a longer period, which allows repeated dosage of the cells and therefore also to invest in some more chronic responses. With the cell system, including ciliated cell as well as mucus-producing cells, it allows the measurement of cilia beating frequency as well as mucus production and therefore also mucociliary clearance. Mucociliary clearance is the main defense mechanism for the upper airways and protects for harmful effects caused by pathogens, but also inhaled particles. For some substances, inhibition of mucociliary clearance was reported, which short-term can lead to irritation and, if persists longer, to a higher chance of airway infections, and long-term to some increased mucus production, which could lead to goblet cell hyperplasia with the chance to develop asthma or COPD. Disturbed mucolytic function is one of the key hazard concerns we have, and we included a lot of our benchmark substances to evaluate the cell system for this endpoint. However, PMRG has not been reported to have an effect in upper airway, and we did not expect it to affect mucociliary clearance in vitro. With the main effect of PHMG exposure in human reported to cause cytotoxicity, oxidative stress, and inflammation in the lower airways, we could not exclude that a certain concentration just also could affect the cells of the upper airways. So our endpoint of concern for PMAG in the cell system is on the barrier function of the cells. Next slide, please. Cells were exposed to PHMG using a Vitro cell cloud chamber on a daily basis for up to 12 days. As mentioned before, one main mechanism for the upper airway is to eliminate foreign particles using mucociliary clearance. While the cells model if cells is capable of mucociliary clearance, it however cannot remove the particles from the Petri dish. So we decided actually to include this mechanism in our experimental design manually, which you can see on the left side. Mucociliary clearance is reported to happen relatively fast, and therefore a daily exposure duration of 30 minutes was chosen, after which the cells were washed and which could recover for another 23.5 hours before biomarkers were measured or the cells were dosed again. Biomarkers were at least measured on day 1, 7, and 12 of the treatment. To study dose concentration response, we added 3 concentration for the 30-minute exposure. In addition, we also choose one concentration to have a slightly longer exposure of 6 hours as a conservative approach, assuming that some particles can't be eliminated as quickly. And as you can see, this actually in the end leads to a quite complicated experimental setting, and therefore ask for some bespoke data analysis. Can you move to the next slide, please? By the mathematical model we normally use allow for dose-dependent response analysis, which is quite similar to the BMD model Marie presented. They do not include time dependencies. So we decided to use a mathematical model, which we developed in parallel in-house for our data analysis. This model uses a systematic approach, and the model itself is trained by including control data from different time and possible interactions of different endpoints, fitting the model in a probabilistic manner, which allows to account for experimental as well as biological uncertainties. Using this model, the 95% credibility range for the controls here presented in pink, as well as for the treated data here presented in black, can be predicted. The model is still under development, so these are just some preliminary results. For treatment with PHMG, only a minor response could be detected. This nicely aligns with what happens in vivo. Some slightly higher release of some cytokine markers, here IL-6 and IL-8, can be detected, but only after 6-hour daily treatment and only after 5 to 6 days of treatment. In contrast, for the 30-minute daily exposure group, no inflammatory response could be detected, but some mild change in tissue integrity were measured. Here, please see that the tissue integrity is not going down, so this is not cytotoxicity, but probably related to some blockage in ion channels. These results show that repeated daily exposure as well as exposure duration can have a big impact on the experimental outcome, and therefore probably also on the risk assessment side. So this is something we need to investigate a little bit further and to find best practice. Next slide, please. For the lower airway, our major hazard concern for the proof of concept study was cytotoxicity and inflammation, which could lead long-term to airway remodeling and fibrosis. So PHMG is actually an optimal benchmark substance for this. As a cell system, we choose the epialveolar system for MATIC, which includes primary human alveolar epithelial cells, pulmonary endothelial cells, and monocyte-derived macrophages. These cells are grown as well on the air-liquid interface and are viable for up to 21 days. One important point for us here was that actually we have a co-culture of cells, since it was reported in literature that these cells respond a little bit more sensitive to some treatments than monocultures. Using TGF-beta treatments, these cells were proven before in a paper from Aerosol et al. To show prophylactic response, where the cells are contracting and have like increased ECM expression. As well, you can see here some myofibroblast formulation with the fibronectin and the alpha small. Since this cell system is relatively new and there's only one paper out there at the moment, we actually spent a little bit more time to examine the morphology and the cell composition of the tissue. You can move on, yeah, thank you. So here on top in the H&E staining, we can see some polygonal epithelial cells on top of the 3 to 4 cell layer, and some spindle-shaped fibroblast cells directly on top of the membrane, and some endothelial cells underneath. Epithelial cell identity can be verified using a pancytokeratin staining, and also fibroblast identity can be verified using vimentin. Using a CD68 antibody, some macrophages could be detected in the endothelial cell layer. So all in all, this actually the cell system looks quite as it is supposed to be. However, we also discovered some unexpected results, as some of the cells show some cilia or microvilli on top of the cells, and we also see some myofibroblast formation in the fibroblast layer. Unfortunately, we couldn't identify surfactant protein C expression of 82 cells in the epithelial cell layer as it was reported before from Aerosol et al. So at the moment, we are not clear if and how much surfactants the cells are producing and how this affects the interaction of the cell with the compound and therefore the downstream toxic responses. Next slide, please. We also investigated a bit on the cell morphology over time, and while we see some tissue signaling over time from a 3 to 4 cell layer down to 1 to 2 cell layer, the cell barrier remains quite stable over the same time, and you could even argue that the tissue at day 12 is probably a little bit closer to the in vivo alveolar tissue. Next slide, please. For the in vitro testing of the alveolar cell system, we followed a similar design as for the Mucilair, and we did some daily exposure of the nebulized PHMG for up to 12 days. Since for the lower airway, the main pathway of clearance is by macrophages and is supposed to be happen within days, we actually decided not to wash the cells before we actually measured the biomarkers after 24 hours or before we have additional dosing of the cells. Biomarkers here as well were measured for day 1, 7, and 12, and we choose like 4 different doses for the treatment. Next slide, please. Analysis was performed in the same way as we done this before. And here the results for each endpoints are shown in plots which allow combined visualization of a dose as well as a time-dependent response. As you can see, PHMG, which is highlighted here, results in loss of tissue integrity already at a concentration of 0.2 microgram per centimeter square, and is already at day 1 of the treatment. This is accompanied by increase of inflammatory cytokines as well as ECM deposition. We think that this might reflect quite nicely what's happening in vivo with PHMG, where this leads to acute interstitial pneumoma, which is characterized by diffuse alveolar damage. For lower concentrations, we also see some increased pro-inflammatory response, which can be observed only after later time points. Next slide, please. As mentioned before, we developed our own probabilistic model for the analysis of the data to adjust for dose as well as time dependency. The model output is used as well for point of departure predictions. When the model is trained on the data, here presented the green dots, multiple parameter samples are generated corresponding to different probabilistic outcomes. This leads to multiple fitting curves representing in the different potential outcome, which is shown here in the light blue and the dark blue. So instead of having one fitting curve, what people are probably normally used to, we end up with a distribution of fits, which can be used downstream for statistical analysis, like, for example, calculating the mean or calculating the 95% probability range, as we showed it before. Calculating now the point of departure therefore also gives not only one value, but also leads to several values. So if you run a fitting curve twice, this way you would see that each time the curve would pass the 95% credibility range of the control data in a slightly different way. Can you click once, please? So this is shown here for the parameter 1 sample, where you can see the actually corresponding dose. And can you click a second time? And this is shown here for the dark blue curve, where you actually can see that the dose is slightly later. So if you would do that now for a large number of time, this would give an estimate of point of departure distribution. And this is what we use for our data. Can you move to the next slide, please? Which is shown here. Point of departure distributions were calculated for all the in vitro data measured, which means all different biomarkers in the 2 cell system for the upper and the lower airways, as well as for the different time points measured. Here is shown only for day 1 and day 12, but we have all the different time points in between as well. Comparing now the day 1 and the day 12 data, you can first of all see that more of the biomarkers respond at the later time point, and therefore only these ones can be used to estimate the point of departure. What you also can see is that the point of departure slightly decrease over time. So it's a little bit lower for day 12 as it is for day 1. And also if you compare it then for the deposition rate, which is shown here as a horizontal lines, you can see that probably the margin of safety will change over time. And this will probably have a big effect on the inhalation risk assessment as well. So for PHMG, maybe this one is not really high, but we could assume that for some other substances, this could have a big effect. The bigger effect here for PHMG is actually mostly for the choice of the cell models we use. So if you compare the upper airways, which is shown here in yellow, to the lower airways, which is the blue and the red points, you can clearly see that this will definitely lead to a different outcome in the risk assessment. Last but not least, we also did a little bit comparison between some interlab and some interdonor variations. And as you can see, while we see here probably the different biomarkers response, we can clearly see that our point of departure calculation would come up in the end with similar results. So this is where we are at the moment with our inhalation risk assessment framework. So we haven't actually calculated the margin of exposure at the moment. And this is where we come to our last slide. Can you move? Which is about gaps and next steps. Maria, are you going first?
Yes, sure. I can give the next steps for exposure. So for the exposure, we still have some uncertainty, and and estimates of regional deposition doses have been talked about, I think, in other webinars, the limited of human data to validate some of the models. We also struggle to incorporate some of the clearance at this time in point, that we can do use the clearance, the MPPD clearance part to do that on the day of the daily exposure. But for chronic repeated dosing, we kind of lack this ready to use to predict a steady state concentration in the lung. So if we were to do this repeated dosing, and how do you incorporate all the multiple clearance mechanisms that we can factor in? So that's something we want to do in the future, and we also will welcome some collaborations to help us with that. We still have some uncertainties in that delivered dose. We use the QCM to track what the targeted delivered dose, but especially over a repeated dosing scenario, how can we make sure that we know what actually the cells are seeing and exposed to? And we also have some plans to compare the MPP outputs with the CFD modeling results, particularly because we are concerned with the hotspots. So we know that it's not evenly distributed, the exposure, the particles are not evenly distributed in the respiratory tract, and there might be some hotspots that we might be missing with MPPD.
For the data generation and concentration response modeling, we then really need to finish off the mathematical model, but we also want to explore it a little bit further. So the model as it's set up at the moment might also be helpful for predict point of departures at times where we don't have actually some measured data. We then have still a lot of other data where we actually need to have a full evaluation on. And I think in the end, we need to have some thinking about how we will make the decision making on that, and probably also probably need to expand the data set a little bit. For in vitro, we have a little bit of uncertainty around exposure duration in vitro. So do we go for 30 minutes versus 6 hours in the upper airway? Is the 24-hour for the lower airway okay? Is probably also the duration in general, the 12 days enough to make a good estimation of the margin of safety. So now at the moment we're measuring quite a lot of endpoints. So I think one thing as well is then for future study to simplify the experimental design a little bit and probably just go to the relevant endpoints. And then we have also large transcriptomic data sets in comparison to this data we already have, which needs to be finalized and completed. And last but not least, we also probably already defined some gaps, so we need to fill. So next for us is looking also on the biopersistency and anti-surfactant inhibition. Next slide. So last but not least, I think this is a really big project. It's a lot of work, and I would like to thank everybody who is involved, which is on the Unilever side, the whole inhalation team. But I also want to thank all your collaboration partners from Epithelium, Charles River Laboratories, and IVS. And as Maria mentioned before, so we have a lot of other collaborations ongoing in the inhalation space, which haven't been mentioned here since there was no part of the talk. But we're always looking for more collaborations. With thanks, I would thank you a lot for listening, and we're open for some more questions.
Thank you, Maria and Iris. As a reminder, we will hold all questions to the end, but please send them in as you have them using the GoToWebinar questions panel, and we will ask them after our next presentation. Our last speakers today are Dr. Sandra Verstralen and Evelyn Friends. Dr. Sandra Verstralen is a biomolecular expert with a background in biomedical sciences. At Vitro, she is the air-liquid interface exposure project leader, coordinating strategic and contract research in this field of respiratory research. She is an expert in the in vitro assay development and validation using human cell models and molecular technologies. Evelyn Friends is an aerosol expert. She received her master's degree in physical geography in 2002 From 2002 to 2007, she worked as an environmental consultant specialized in soil contamination and remediation. Since 2007, she is a scientific researcher at Vitro with expertise in aerosol exposure assessment and generation. Since 2014, both researchers are responsible for the air-liquid interface platform for in vitro inhalation testing at Vitro. Thank you both. We look forward to your presentation.
Okay, thank you for the nice introduction and to set up this inhalation webinar series. So I'm Sandra Verstralen and I will start the talk of today on the INSPIRE project. It is on in vitro inhalation testing of silane and surfactant compounds, and this is a project supported by PETA. So we will start with the agenda. So first I shortly introduce Vitro, followed by the state of the art on inhalation toxicity testing and how we contribute at Vitro in this field. And the main part of this talk is on the case study INSPIRE. We will try to explain in detail the different aspects of the project. So my colleague Evelien, who is aerosol expert, will explain the part starting from the test chemicals and the controls up to the Vitro cell 64 exposure system and parameters, and I will cover the other parts of the talk on the test systems we used, the designs, results, some troubleshooting, and the next steps. So here, Vitro is the Flemish Institute for Technological Research with its headquarters in the green area in Mol in Belgium. It's a strategic research center of Flanders, funded 1/3 by the Flemish government. And on the picture, you see one of the Vitro buildings where we perform our in vitro inhalation studies. Here on the left, some figures of 2020. So Vitro counts around 950 employees and covers 43 nationalities. And the activities of Vitro are organized in 5 teams, focus on sustainable energy, chemistry, materials, land use, and also health. So the in vitro inhalation testing is, of course, part of this health department. So acute toxicity, a first step risk assessment for non-pharmaceutical compounds. You know how inhalation is a major exposure route for humans, respiratory tract, target tissue, and also portal of entry to the systemic circulation. For regulatory purposes, acute inhalation toxicity testing is performed in rats and in mice according to OECD guidelines. These in vivo models are biased by differences in respiratory tract architecture, function across patients, so sometimes it's difficult to draw conclusions on potential health hazard of inhaled compounds in humans. So research efforts focused on developing alternative human relevant models. We call them the non-guideline methods. There are 3 main categories. We have the lung on a chip, and here I want to refer to the talk of Josue Snitman on May 19 of the webinar series. The next category is the ex vivo human precision cut lung slices. So this was presented by Holger and Vivek last week. And then you have the cell cultures, and this is also the focus at Vitro. So to date, none of these alternative models are accepted by regulatory agencies as standalone replacement for inhalation toxicity testing. So at Vitro, we offer expertise in alternative methods for inhalation testing using the air-liquid interface cell cultures. So you probably know that the cells are cultivated on an insert. You see it on the picture below of the slide. They are fed from the basal side with medium, and they are in contact with the air on the apical side. So we have air-liquid interface expertise for simple monoculture cell lines, B2B, for example, Calu3 A549. We have expertise in the 3D tree-culture alveolar model developed by the Luxembourg Institute of Science and Technology. And we also work with 3D tissues from human donors from epitheliums. And these air-liquid interface cultures, they are used a lot for respiratory research, and exposure is mainly done by applying a liquid to the apical side of the cultures. But to simulate more the in vivo situation, we have at Vitro a platform for realistic gas, vapor, or aerosol exposure. So on this slide, you see the platform we have at Vitro. So we have 4 different exposure systems, 2 commercial Vitro cell systems with a perpendicular direct flow, 1 system with mist generation, well known as the Vitro cell cloud. It was also mentioned in the previous talk. And we also have 1 in-house co-developed system with a laminar flow. It's called Navetta. And for the Inspire project, for this talk, we use the VitroCell 6.4 direct flow system. And next to exposure systems, we have also equipment for wet and dry generation. For wet generation, we have different atomizers, nebulizers. And for dry generation, we work with precise inhale on the bottom left and with the rotary brush generators. And the precise inhale is recently introduced to the platform and can also be connected to DPI and MDI inhaler devices. Now we will explain in detail how we use this platform for testing the silane and surfactant compounds. So this project is called Inspire, in vitro system to predict respiratory toxicity, supported by PETA. The goal is to demonstrate the usefulness of different in vitro test systems and predict the ability of chemicals to cause effects on human respiratory tract. So in 2019, we performed a proof of concept study, phase 1 study, it's in the table. This was with using 3-ethoxysilane in a B2B model. The data were presented last year in a webinar. You can see here also the link of the talk of last year. End 2020, we started with phase 2 as a follow-up study in the same model in the B2B cells. And you see in the table some key differences from phase 1. So, for example, we reduced exposure time, we added additional compounds, we added a true negative control, we changed the carrier gas, we did post-exposure but not submerged, but at the air-liquid interface, to name a few. So this table will be shown again later on in the presentation when we will present some preliminary results. And then we also have a phase 3, and in phase 3 we use the 3D Mucilair model. And when using this model, we can add some additional endpoints, and we also added a 7-day recovery period. So the phase 2 and the phase 3 studies are still ongoing due to some COVID-related delays in delivery of consumables. So respiratory research and the use of these ARI cultures boosted since last year. So that's also a positive aspect of the pandemic. So I will now switch to my colleague, Evelien, who will zoom into detail into the next phases of this project.
Yes, okay, thank you, Sandra. So I will continue with a small description of the test chemicals that were used. So we used silanes and surfactants. Silanes were selected because in vivo data, they show acute toxicity by inhalation. And also silanes hydrolyze quickly and form ethanol, methanol, and hydrochloric acid, which are known to have toxic effects. Silanes are used for corrosion protection and adhesion promotion, and also used for surface modifications. The 2 different silanes that we used were 3-ethoxysilane, which is a category 2 chemical, and 3-methoxysilane, which is a category 1 chemical. The next group we use in our ELI platform is the surfactants. 2 different types of surfactants were selected, which were oleoyl sarcosine and Triton X100. They were selected because the EPA is also performing research with these compounds, and they may pose an inhalation hazard to human when they are inhaled. So the oleoyl sarcosine is an anionic surfactant with a category 4, and the Triton X100 is a non-ionic surfactant. The oleoyl, sorry, Sandra.
Sorry.
The oleoyl is used as corrosion inhibitor in aerosol products, and the Triton X is often used in laboratory as a chemical to lyse cells to extract protein and organelles, or to permeabilize the living cell membrane for transfection. Next slide, please. So next to the compounds, we also selected negative and positive controls. The negative controls that we used were clean air and nitrogen in the first place. The selection depended on the compound that was used. Clean air was retrieved from a compressor, which was double HEPA and active carbon filtered, and nitrogen was retrieved from a storage tank. Also, as a negative control, a 0.9% sodium chloride solution was atomized, which is shown in the picture below. As a positive control, nitrogen dioxide was used. It was from a gas cylinder, and nitrogen was used for dilution, and the diluting element is shown on the bottom. We used 25 ppm for the B2B cell lines and 800 ppm for the Mucilair. For comparison, the outdoor concentrations for nitrogen dioxide for EPA is 53 ppb, and for the EU it's 40 ppb. So it's really high levels that we needed to get a positive control. Next slide, please. So now I will show some pictures about the generation setup we used. So first, the generation setup for silanes. This was based on the capillary dosage system that was developed at Vitro by Muller, with the paper reference shown below. I will briefly guide you through how it works. So on the analytical balance, there is a canister in stainless steel, and there are two capillaries attached on the lid of this recipient. On one side, the capillary is attached to nitrogen. The recipient is filled with the silanes, and then a pressure is put on the recipient, and the silane flows towards the heating elements that is shown in at position 4. There, within the heating element, there's a cotton clog situated. There the silane will be evaporized, and with additional of extra nitrogen, the silane is transported to a glass distribution line, which I can show on the next slide. So here you can see the system in total. On the left side, the small picture that I showed previously with more detail about the cotton cloth and the temperature part. So I colored the lines to give more detail. So on the yellow line, the nitrogen is injected into the canister. On the orange line, the silane is transported towards the heating element, where it's mixed with nitrogen. Then the evaporated silane is transported with the red line towards a flow splitter. The flow splitter was not used in all cases. That was dependent on the concentration we needed. And afterwards, after the flow splitting, the evaporated silane was inserted with the green line into the glass distribution line with an extra dilution on the left side. On the blue circle in the middle, you can see the mixing of the test compound with the carrier gas. And then the long glass, and at the end of the long glass distribution line, you can see the connection to the Vitro cell SIG4 module. So on the small picture on the right-hand side, you can see the Vitro cell model where it's opened. So you can see the 4 positions where 4 inserts are positioned. And each of those 4 positions are have a separate extraction of a flow of 3 milliliters per minute. So next slide, please. So this is the exposure setup for the Triton X100. It's in the black box in the black arrow. So we used a different atomizer for this setup. The Triton was diluted with molecular water to prevent foaming of the liquid inside the atomizer, and also because we noticed, or we found a dose range of smaller than 2.5 for the B2B cell lines and smaller than 10% for the Mucilair. So on the black dot on the glass atomizer, pressurized clean air was put, and then the droplets, the Triton droplets, were transported towards the distribution unit and mixed with additional dilution air depending on the concentration that was needed. And also here on the right-hand side, you can see the Vitro Cell 6.4 modules and a slightly different distribution unit than for the previous exposure setup. So here it's in stainless steel, but again, the 4 positions connected and each of the positions sampled separately with 3 milliliters per minute from the main aerosol flow line. Next slide, please. Then the exposure setup for the oleo wheel, it looks similar to the previous one, only we needed to use a different atomizer for this exposure because of the viscosity of the oleo wheel. We also had to dilute it with 10% of ethanol to get it in the right concentration and to get it into the atomizer to get the right droplet size. We also diluted this at the atomizer itself. So it's the second Teflon tube on the back. And with this, we obtained oleo wheel concentration of 188 until 650 micrograms per cube. And that was used for exposing the Mucilair. Next slide, please. So for the aerosol characterization and the dose determination, we use different techniques. So for the silanes, we looked at the stability of the silanes in the carrier gas with an online monitoring with an FID analyzer, which is shown on the photo. And with this data, also the generated concentration was calculated. The dose was determined by exposing membranes with cells to the silanes, and after exposure removing them with a scalpel blade and storing them in a 50 milliliter tube at -20 degrees C. Those membranes will be analyzed with ICP-AES, but it still has to be performed. For the oleo eel, we determined the mass concentration by simply sampling on a 25 millimeter quartz fiber filter and weighing the filter before and after the sampling. And together with the sampling volume, we calculated the mass concentrations in milligram per cube, which the data was shown previously. For this exposure, we determined the dose by taping dry inserts. And after the oleo wheel exposure, this membrane was rinsed with 100 microliters of ethanol. And this ethanol was collected for LCMS analysis. For the Triton X, we are going to perform the dose determination also, and it will be in a similar way as for the silane. So we will seed some cells on a membrane, expose it to Triton X 100, and remove it with the scalpel blade and also store it in the freezer. Next slide, please. So here, there's an overview of the 2 analytical systems that we have at Vitro for this analysis. So for the silanes, we will use the ICP-AES to determine the silane concentration on the membrane. This is performed by digestion in TMAH. And for the surfactants, LCMS will be used, and this will be used for the taped inserts that were rinsed with ethanol, but also will be used for the membranes with the cells on it. So here in detail, the 6-4 system, which I showed previously also in the different exposure setups. So, so for each position, we used a flow rate over the cells of 3 milliliters per minute. The flow rate in the system elsewhere, that will depend on the concentration and the dose that was needed for each exposure setup. We set the trumpet height at 3 milliliters. And this module is thermostatized from the bottom while it's connected to a water bath set at 37 degrees. We also used nitrogen for the exposure of the silanes because of the reactivity. Next slide.
Okay, thank you, Evelyn, to explain in detail the generation and the exposure setup. So I will follow with the test systems we use and some parameters and endpoints. So we use 2 in vitro test systems, and one is Beast2B. It's a 2D normal human bronchial epithelial cell line. In the table below, you find some key parameters for the air-liquid interface protocol. So cells were seated on 6-well sigma inserts with a pore size 0.4 and a density of 50,000 cells per square centimeter. For the protocol, the growth protocol, the cells are first grown submerged for 48 hours, and at the day of exposure, the apical medium is removed. And also the bronchial epithelial growth medium is exchanged for bronchial epithelial basal medial to stop cell growth. And during the culture of the Beast2B, we also removed the bovine pituitary extract from the medium to avoid animal-derived products in culture medium. And this was tested before, not at Vitro, but at the Luxembourg Institute of Science and Technology. So in vivo, silanes may lead to some key events, for example, cellular death, loss of epithelial barrier, inflammation. As explained by Evelyn, the surfactants, they also disrupt cellular membranes. So we tried to translate these in vivo key events to some in vitro endpoints. And you see it here on the slide. So we checked for cell viability with, we used the Presto Blue protocol. So it measures metabolization activity. We also checked cytotoxicity, which is reflected by release of LDH with the Cytox-1 assay. And we also measured some inflammatory markers here, IL-2, 6, 8, and TNF-alpha. And for this, we have a mesoscale discovery platform. Another test system which is included in the project is Mucilair. So this was already explained in the 2 previous talks. So we work here with normal bronchial monodonor tissues with different ages and gender. The tissues are produced by Epithelix using a standardized platform, and we apply as much as possible the Epithelix protocols for the maintenance and the readouts of the different endpoints. Maybe important to mention here is also that we washed the tissues before 24 hours before the exposure, and we also did a basal tear measurement at that time point. So we checked the same endpoints as was previously shown for Beast2B, but we also included some additional ones because we have here a 3D model, so we can check barrier integrity, the tear measurements. We can check cilia beating. For this, we use the SAVA system. And also morphology will be checked, but that is not done at Vitro, but at Serba research. Another part of the experimental design here of the silanes. For the exposures, we perform a 30-minute exposure, and on the right, you see the different conditions for the air exposures. So we have the carrier gas, nitrogen. We have a true negative control sodium chloride. It's an aerosol exposure. We have the positive control nitrogen dioxide, which is a gas exposure. And then we have the 2 silanes in 3 different concentrations, 3 ethoxysilane and 3 methoxysilane. And this is a vapor exposure. Then we also included some liquid exposures for the positive controls of the assays. So we have a positive control for the LDH readout. This is delivered together with the kit with the assays and is Triton X. We use LPS as a positive control for inflammatory response, and we use isoprotenolol as a positive control for cilia beating. And then next to all these conditions, we also have just the incubator controls, and these are just the cultures that are not exposed and just stay in the incubator for the same period as the other conditions. This was for the silanes. Here a bit more for the surfactants. So for surfactants, we have the carrier gas clean air, again, sodium chloride as negative, nitrogen dioxide as positive, 3 concentrations of Triton X and 3 concentrations of oleolyl. And these are aerosol exposure of the surfactants. But next to the aerosol exposure, we also want to compare with the liquid exposure technique. So we expose the in vitro test systems to the concentration apical exposures by adding a liquid on the apical side, and we compare against the solvent controls of both surfactants. Again, also some positive controls of the assays that are included, and of course the incubator controls. Here the experimental design, an overview of the readouts in the B2B for silanes and surfactants. So this was already mentioned that we have 4 replicates per condition in the VitroCell 6.4. We do 30-minute exposures. We also include an air-liquid interface post-exposure time. This was not done for the chemical analysis. We include a 30-minute post-exposure for the LDH readout, and up to 24-hour post-exposure for all other readouts, LDH, Presto Blue, and the inflammatory markers. And then the numbers in the tables, they reflect then the different tissues we used for the different assays. And here the same for Mucilair in the 3D system, 30 minutes exposure, no post-exposure for the chemical analysis, no 30-minute post-exposure. We have an up to 24-hour and a 7-day recovery period for all other endpoints. And then the numbers reflect the tissues we have available for the different readouts at the different time points. On the right, you see the Vitro cell, and you see that we need to use adapters because the Mucilair is not in 6 well but in 24 wells. So we changed the system with adapters for the 24 well size. And then for B2B and Mucilair, we will run at least 3 valid independent biological experiments. So we will obtain a lot of data when having all these studies done. So in the coming slides, results of the different project phases, we have 3 phases, will be presented. We start with an overview of results from the completed phase 1 study in B2B cells exposed to 3-ethoxysilane. The table shows the nominal concentrations that were used and also the amount of 3-ethoxysilane which was measured onto the cells, and the delivery efficiency, which is dependent on the exposure system and on the physicochemical characteristics of the compounds, was calculated and was 14%. On the next slide, we have an overview of cell viability done by Presto Blue. We see here the mean change in viability of the B2B cells after 1 hour exposure to 3 concentrations of 3-ethoxysilane vapor. Different colors represent the 3 independent biological experiments with 3 technical replicates for each condition per experiment. And on the y-axis, you see the percent cell viability compared to incubator controls, which is set to 100%. And then on the x-axis, you see the different conditions, which was already explained in the experimental design. And here we see a significant dose-dependent decrease in viability after exposure to 3-ethoxysilane vapor as compared to the incubator and also clean air controls. The results were statistically significant for the middle and the highest concentration, and also the positive control was also significantly decreased for cell viability. Here on the slide, the results of the cytotoxicity outcome reflected by measurement of LDH release. So again, a significant dose-dependent increase in cytotoxicity was observed after exposure to 3-ethoxysilane vapor. Again, also the positive control showed a significant increased mean cytotoxicity. Besides cell viability and cytotoxicity, also the pro-inflammatory response was checked for the 2 lowest concentrations of 3-ethoxysilane vapor. And here on the figure, you see a statistically significant release of IL-6 for the highest concentration of 3-ethoxysilane. And the same trend was observed for the other pro-inflammatory markers, IL-2, 4, 8, 10. You see it on the slide. And 2 markers were below detection, IL-1 beta and interferon gamma. And also LPS was here used as positive control and induced also a statistically significant release of all the markers compared to the incubator controls. This was a study of 2019, the phase 1 study. And the data obtained here in 3 independent experimental runs indicated that the air-liquid interface exposure method in bees2bee was robust and reproducible. So this is of course important as a start for maybe validation purposes later on, and also as a start for the phase 2 in 2020. The phase 2 is the follow-up in bees2bee, but after some discussions with stakeholders, we slightly changed the design. You see it here in the table, some key differences. So we reduced the exposure time because we added some extra compounds, and by reducing the exposure time, we can do more runs in 1 day. We added the 3-methoxysilane and also both surfactants. We also added a true negative control, sodium chloride. We changed nitrogen as carrier gas for the silenes because 3-methoxysilane was even more reactive than the 3-ethoxysilane. We only tested 4 cytokines instead of 10. We did a post-exposure at the air-liquid interface and not submerged, and we removed during maintenance of the bees2bee the bovine pituitary extract from the media. And then for phase 3, this was shown. We work with the Mucilair model. We apply the same compounds, and we added some additional endpoints and also the 7-day recovery period. So phase 2 is still ongoing, so we will present some examples of individual experiments. This is not shown, but first we always start with some range finding studies, some determinations of the doses, which was explained by Evelyn. We needed to redo the range finding studies for 3-ethoxysilane because we slightly changed the study design. In the figure on the left, you see the mean change in viability of bees2bee after 30-minute exposure to 3 concentrations of 3-ethoxysilane, 150 and 150 ppm. This is for 1 final run, and then for each condition, you see then the 3 technical replicates on the figure. On the y-axis, cell viability is shown compared to incubator control, and here the nitrogen carrier gas was set to 100%. And on the x-axis, you see then the different conditions, also clean air and sodium chloride, and the positive control nitrogen dioxide is shown on the figure. You also see that the positive control showed a significant decrease mean viability compared to both negative controls. The same can be concluded for 3-methoxysilane vapor on the right, the figure, and the following concentrations were used for one run, 1, 25, and 85 ppm. Here again, an example of one study of cytotoxicity for both silanes, so reflected by measurement of LDH. You see a dose-dependent increase for 3-ethoxysilane vapor, and also the positive control showed some increased cytotoxicity. And on the right, you see for the 3-methoxysilane vapor with only for the highest concentration an increase in cytotoxicity. On the next slide, for the surfactants, it was explained in the design. We apply both exposure techniques. On the one hand side, the liquid exposure, and we compare with the aerosol exposures. But here we show some preliminary data, on the liquid exposures. So in the figure on the left, mean viability of Bs2Bs exposed to 3 concentrations of Triton X100 compared to the solvent control, we see a decrease in viability. And for oleolyl on the right hand side, also a dose-dependent decrease. And maybe here the 50 micromolar is even too high because there is no viability anymore. And this is also reflected in the cytotoxicity assay on the next slide. If we look to the next slide, and then the right figure here for the 50 micromolar, we didn't pick up LDH anymore because tissue was already dead. But here for the middle concentration, an increase in cytotoxicity was observed. And for Triton, we saw it for the highest concentration. For comparison, the highest nominal concentration of Triton X100 for the aerosol exposure is 2.5%. So there is already a difference in concentrations used for both exposure methods. So we also want to share some troubleshooting with you. First, we focus on the Bs2B system. We have some issues with LDH readout at 30-minute post-exposure. So I mentioned it in the design. We do 30-minute post-exposure and up to 24-hour post-exposure for LDH. And for the 30-minute, we have some issues. We use the Cytox-1 assay, which is fluorescence-based. We know half-life of LDH is approximately 9 hours. We presented the design, and so it's clear that there are a lot of handlings to do at the day of the exposure. So this means that it's complex to combine it with LDH assay and readout at 30 minutes post-exposure for all conditions, since you need to expose cells to new condition every 30 minutes as well and collect all samples in between. So we had an idea, we learned it from another project, to collect and store the basolateral samples at -20 and perform the LDH the next day, but yeah, this was not working. We kept the samples at 4, but again, this was also not working. So now we decided to perform the LDH at the same day of exposure with extra help of some technicians. And we are also checking another protocol, which is based on absorbance instead of fluorescence. Another thing is maybe good to mention here that we also excluded one run because the trumpets in the Vitro cell system were adjusted with the wrong inserts. So we know from previous project that even a small difference in the height of the trumpets above the cell layer can influence the amount of compound reaching the cell surface. So This was a small mistake in the lab, but we needed to exclude this. But it's good, I think, we mention this here at this platform, because every detail is important to obtain valid, reproducible results with these aerosol vapor exposure techniques. The next slide, we also have a little bit troubleshooting with the 3D system. Evelyn mentioned it already. Historically, we used the nitrogen dioxide as a positive control in-house, but we need to apply a very high dose of nitrogen dioxide for the Mucilair to see effects. So we also see responders and non-responders. So we need to look into this data, check perhaps a mechanism of nitrogen dioxide exposure. Maybe this is donor dependent, we don't know yet. So if there are suggestions from the audience for good positive controls for this system, test system, aerosol, gas or vapor positive controls, we are happy to hear. And then another reason maybe for variability might be the quality of the mucus, but yeah, we don't know yet, so we need some more runs to draw conclusions on this. So finally, we approach the last slide. Next steps, and we will start with fixing the troubleshooting. We want to obtain at least 3 independent valid runs in both test systems for the silanes and for the surfactants. So we will obtain a large data set which allows us to determine if the test systems can detect decrease in toxicity that correlates with increasing carbon length for the silanes, which is not evident right now from available animal data. We can compare the liquid exposure method and aerosol exposure method for the surfactants. We can determine advantages of using the 2D B2B model versus the 3D model Mucilair. The goal is also to do in vitro to in vivo extrapolation, and we are open to ideas as how to do this. We are conducting the chemical analysis, so we can determine the cellular doses and possibly link it to the observed changes in the endpoints. So we really want we really hope that this data set help us to move forward in this domain in inhalation toxicity testing. So this work was sponsored by PETA. Now for the next slide, yeah. We really want to thank them for the nice and open collaboration, their expertise in the field, critical review, and really positive, fruitful discussions on this topic. It really helps us to move forward in this domain. At Vitro, we work in a multidisciplinary team. I hope you saw it from the previous slides that we need different expertises to do these experiments. On the picture, you see the core team, but of course, we get a lot of support from other colleagues, which are mentioned below the picture. Also, Sven Verkouteren is our business developer, and he supports us to promote this platform not only for inhalation toxicity testing, but also to use this for pharma and biotech applications. So we are always open for collaborations in this field. So I hope you enjoyed listening to this case study, and I want to thank you for listening.
Thank you, Sandra and Evelyn, and thank you to all the speakers. Looking at the time, we have 2 minutes left, so unfortunately the Q&A session is falling short, but I think we can do a quick set of 1 question per speaker, and we will make sure that all the other questions will be answered in writing after the webinar. For Marie, a couple of questions came in as to why you used liquid exposure instead of using systems like an ALI exposure system. Can you comment on this, Marie?
Hi, Andy. So yes, I can provide a brief comment. So I believe the Mucilair assay was chosen based on various or constant discussions between Charles River Laboratories, because this work was done at that particular facility at the time, and the Mucilair assay was deemed as the optimal in vitro model instead of using the air-liquid interface.
Thank you, Marie. One question for Maria and Iris about whether you could please discuss whether you had any input on these models from regulatory authorities such as the EPA or from European regulatory authorities, whether you're working with them to make sure that they accept your approaches and validate your assumptions.
Sure, I actually answered as well on the chat. So we haven't not yet engaged directly on this topic on inhalation toxicity with regulatory agencies, but we have several activities. We have a CRADA with the EPA where we discuss NAMs for systemic toxicity, and we also discuss in general some of the other approaches. And we're part of several trade associations, well, part of the European Trade Association, where we have regulatory interactions as well within the regulators in Europe or other activities as well, but not directly on our assays, but we are hoping to increase that engagement in the future. But on general NGRA, we have engaged with EPA and some of the European regulators.
Thank you, Maria. And then one last question for Sandra and Eveline. If clean air control exposures are causing significant adverse effects, as we have seen in the toxicity or cell viability data, doesn't that mean that the experimental setup is not appropriate? The cells are compromised and you may get false responses because of that?
For clean air and nitrogen, for the carrier gas, we have a criteria that the viability must be above 85% viability to use the data. So for sure, if there is more decrease in viability, we exclude the run. Then we cannot use it. That's for sure. That's a control. So we need to keep an eye on all controls included in the study. But most of the time, the clean air nitrogen data are above 90%, but sometimes we have 3 replicates and sometimes one is a bit lower, but yeah, we do different independent experiments as well. So yeah, we keep an eye on that and only include the relevant data.
Thank you, Sandra. And thank you everyone again for joining today's webinar, and to our speakers. Our 3-part inhalation webinar series comes to an end, and I hope you enjoyed listening in. If you have missed any of the previous webinars, please make sure to check out the recordings and the slides at the website shown at the bottom of the screen. Thank you again for listening in, and enjoy the rest of your day. Goodbye.